In the previous session, we discussed the biological basis of drug resistance, bacterial persistence, and the evolutionary principles underlying drug-resistant tuberculosis. Today, we will build on that foundation by correlating these concepts with the mechanisms of action of individual anti-tubercular drugs. Understanding the drug target is the first step towards understanding how resistance develops. Let's now examine the molecular mechanisms of resistance, one drug at a time, beginning with the key first-line antitubercular agents like rifampicin. Tuberculosis continues to be one of the leading infectious causes of morbidity and mortality worldwide. Despite the availability of highly effective chemotherapy, the emergence and transmission of drug-resistant tuberculosis, particularly rifampicin-resistant and multidrug-resistant tuberculosis, remain major global public health challenges. Among all first-line antitubercular drugs, rifampicin occupies a unique position. It is not only one of the most potent bactericidal agents against MTB, but also the drug primarily responsible for the remarkable success of modern short-course tuberculosis chemotherapy. What makes rifampicin particularly fascinating is that its activity depends on an exceptionally precise interaction with a single bacterial agent. Consequently, a single nucleotide substitution within a single bacterial gene may be sufficient to abolish drug binding, rendering one of our most effective antitubercular agents virtually ineffective. This illustrates one of the fundamental principles of antimicrobial resistance. Small molecular alterations can produce profound clinical consequences. Therefore, understanding rifampicin resistance is far more than an academic exercise. It provides the biological basis for interpreting molecular diagnostic assays, selecting appropriate treatment regimens, understanding multi-drug resistant TB, and ultimately improving patient outcomes. During this lecture, we shall examine the normal mechanism of action of rifampicin, the structure and function of bacterial RNA polymerase and molecular significance of the RPOB gene and the rifampicin resistance determining region. The structural consequences of resistance conferring mutations, and finally the laboratory methods currently used to detect these mutations in clinical practice. Rather than memorizing isolated facts, I encourage you to approach this topic as a sequence of interconnected molecular events. Once the biological principles become clear, the mechanisms of resistance become intuitive and much easier to understand. With that background, let us begin by answering a fundamental question. Why is the rifampicin considered the backbone of modern tuberculosis chemotherapy? That is why it is in front line against TB. To appreciate the significance of rifampicin resistance, we must first understand why it is the most important drug in the treatment. So, by the end of this lecture, you should be able to describe the molecular mechanisms of action of rifampicin and explain why it remains one of the most effective first-line ATD. Rather than simply remembering that rifampicin inhibits RNA polymerase, our goal is to understand how this interaction occurs at the molecular level and why it results in rapid bacterial killing. Secondly, you should understand the structure and function of bacterial DNA-dependent RNA polymerase, with particular emphasis on the beta subunit, which serves as the molecular target of rifampicin. Appreciating the structure-function relationship of this enzyme is essential for understanding the mechanisms of antimicrobial resistance. Our third objective is to explore the genetic basis of rifampicin resistance. We will examine how spontaneous point mutations within the RPOB gene alter the architecture of the rifampicin binding pocket without significantly impairing the catalytic function of RNA polymerase. This elegant molecular adaptation enables the bacterium to survive despite continued exposure to the drug. We shall also discuss the rifampicin resistance determining region RRDR, an 81 base pair highly conserved segment of the RPOB gene that harbors the vast majority of clinically significant resistance conferring mutations. Understanding the biological importance of this region is fundamental to interpreting modern molecular diagnostic tests. Another important objective is to correlate molecular microbiology with lab practice. We will review the principles of expert MTB RIF Ultra, TrueNAT MTB RIF DX, line probe assays, and whole genome sequencing, with particular emphasis on what these tests detect, how they detect resistance, and the limitations that every microbiologist and clinician should recognize. Finally, we shall translate these molecular concepts into clinical practice. We will discuss why rifampicin resistance is considered a critical marker of multi-drug resistant tuberculosis, how rapid molecular diagnosis influences patient management, and why understanding resistance mechanisms is essential for effective antimicrobial stewardship and global tuberculosis control. By the end of this session, my expectation is not merely that you will remember a list of mutations, but that you will understand the biological principles underlying rifampicin resistance and confidently apply this knowledge while interpreting laboratory reports, managing patients, and teaching future generations of healthcare professionals. With these objectives in mind, let us now address a fundamental question. Why is rifampicin regarded as the backbone of modern ATD therapy, chemotherapy, and what makes this drug so indispensable in the treatment of tuberculosis?